Alright, ladies and gentlemen, this is going to be our next video on molecular biology. What we're going to be doing is reviewing DNA. Let's get started on this next video. So let's go through and review DNA. DNA is deoxyribonucleic acid, and it's a self-replicating material which is present in nearly all living cells as the main constituent of chromosomes. And it's really our carrier of genetic information. So if we're thinking about what DNA is, well, it's the genetic blueprints for an organism. It gives the directions to enzymes to go through and assemble our proteins to allow for that organism to exist. So DNA is going to consist of three parts that we've already learned about. Remember, DNA nucleotides consist of a phosphate. They're also going to consist of a pentose sugar, and then they also consist of our nitrogenous base. Now, all of these are going to come together. All of our nitrogenous bases form our double helix, and they're held together by these hydrogen bonds. So if we think about this, each of those nucleotides, remember they're distinct, they all form together, they form that double helix, and that's what's read by enzymes to go through and create organisms. So if we're thinking about this hierarchy of DNA, we've learned about this a little bit more, but let's try to make sure we understand. The smallest part of DNA, or the first part, is nucleotides. We assemble those nucleotides into that double helix, and that double helix is what we call DNA. Now that DNA is very, very long, so we have to wrap it around these special proteins called histones. And when we go through and wrap our DNA around these histones, we call it chromatin. And this chromatin is spread out. Parts of it are wound around the histones, and then parts of it are kind of uncoiled a little bit, so that way enzymes can go through and read the genetic information. Now, when we go through cellular replication, we're going to go through and coil those histones really, really close together. And when we coil them all close together, the DNA is going to be wrapped around each of these histones very, very tightly. It's going to give it this X shape, or this chromosome. And this chromosome is supercoiled, so that way when we go through replication, it's very, very easy and simple for the cells to go through and separate out that genetic information. So we have nucleotides, which is the smallest part, chromosomes, which is the largest part, and then everything in between. So we have to understand the research by Erwin Chargaff. And Chargaff went through and found that there's equal amounts of adenine and thymine and equal amounts of cytosine and guanine in organisms. And what he did was he took yeast, beef spleen, bacteria, put them in a blender, ground up that organic material, and when he went through and ground up the organic material, he was able to look at each of the nucleotides. And what he found was that each of these nucleotides, the A's and the T's, the thymines and the adenines, and then the G's and the C's, the guanine and the cytosines went through, and they were in equal proportions. So what he came up with was Chargaff's rule, and this rule states that that there are equal amounts of A to T and G to C. So from Chargaff's rule, what we can see is that our A's and our T's are going to match up to each other, and then our G's and our C's match up to each other. So if we look at this, what we get is hydrogen bonding that occurs between the G's to C's, the T's to A's, the A's to T's, and the C's to G's. Remember, hydrogen bonds hold that double helix together. And then what we get is strands of DNA that are anti-parallel. Now, what do I mean by anti-parallel? Well, one strand is going to line up in one direction. The other strand is going to line up in the other direction. So what we see here is our G's, our C's, our A's, and our T's match up. They're hydrogen bonded together. But when I say anti-parallel, the strands of the DNA double helix are going to occur in opposite directions because of the opposite orientation of the sugar molecules. So if I were to take that orange strand of DNA and flip it around 180 degrees, well, they would be exactly the same as the blue strand. However, because our A's and our T's have to go through and match up, we call them anti-parallel. The orientation of both of those strands is the reciprocal, or the opposite. So what do I mean by anti-parallel? Well, think about this. The orientation of the DNA is opposite. So let's forget the A's and the T's and the G's and the C's. Purple's always going to go with blue. And then if we look at this, orange is always going to go with green. Now we're going to get our anti-parallel strands of DNA. We have our strand on the left, and then our strand on the right. Purple is with blue, orange is with green, green is with orange, and blue is with purple. They're perfect, they're lined up, they're held together by hydrogen bonds. Now if I take this strand on the right and I flip it over 180 degrees, we can see it's a perfect match. The orientation is opposite, it's anti-parallel. However, it's a perfect match of the other strand. So we've been talking about hydrogen bonding, but why are hydrogen bonds important? Remember, a hydrogen bond is a weak bond due to polar interactions between the molecules. So when we go through replication, an enzyme is going to go down the middle of this DNA, and what it's going to do is separate those two strands equally. Well, this is important because the weak bonding of these hydrogens allows the DNA to uncoil, so that way we can replicate it. If we had really strong covalent bonds, it would take a lot of energy, and it would risk mutations in the DNA occurring. Hydrogen bonds are relatively weak intermolecular forces compared to covalent and ionic bonds, so therefore we can separate that DNA easily. Now if we're continuing upon this, well, this is important for replication. During replication, we have one strand of DNA, and during our synthesis phase of either mitosis or meiosis, we need to duplicate that DNA. So we can go through and get an enzyme to break those weak hydrogen bonds. We can separate out that DNA, and then what we can do is add two complementary strands with enzymes, and as a result, what we're going to get are two new strands of DNA. So we had our original strands, and then we added two strands onto each of those two sections of DNA, and now we have two exact copies of DNA that can be distributed out into two different cells. So, did you guys learn? Well, did you learn a couple of things? Did you guys review nucleic acids? Did you guys go through and review parts of DNA, such as the nucleotides, the chromatin, chromosomes? And then did you go through, learn the hierarchy of that DNA, how they're related? And lastly, were you introduced to replication, because that's going to be the next topic of discussion in the course. This will be the end of the video. I will see you all in class tomorrow.